Let's talk about nucleic acids. Nucleic acids are important macromolecules composed of the 5-carbon sugar, a phosphate group, and a nitrogenous base. Here you can see the phosphate group. It's connected to a 5-carbon sugar. You can see the 5-carbon sugar right here. And you can see that there is a nitrogenous base right there. This is the nitrogenous base. So if it's a DNA, then the sugar is a deoxyribose. And if it's an RNA, then the sugar is a ribose sugar. To the left here, we see a deoxyribose. Notice that you have a hydroxyl group here and a hydrogen group here. On the other hand, we have a ribose, which has 2 hydroxyl groups. So the reason we call this a deoxyribose is because it's missing one hydroxyl group. So whereas in the ribose, there is no missing hydroxyl group there. So deoxy is for missing an oxygen. Deoxy, here you don't have a hydroxyl group because you're missing an oxygen. And the function of nucleic acids is to create, encode, and store information within cells of all living organisms. It makes up the composition of the genetic material. Now, nucleic acids form helical backbones. DNA forms 2 helices and RNA forms 1 helix. Here you can take a look at a picture of the RNA. The RNA forms 1 helix, and I'm labeling the helix right here, whereas DNA forms a double helix, and you can see that you have a double helix going up. One starts here, another one starts right there. The reason DNA forms double helix is because of the hydrogen bonding between nitrogenous bases. Recall, nitrogenous bases can sometimes just be called bases, and these are one of the 3 components of the nucleic acids, as we said, whether it's DNA or RNA. Interactions between the bases is responsible for the helical structure of DNA and RNA. The nitrogenous bases are adenine, cytosine, guanine, thymine, and uracil, and they're abbreviated by the following: A for adenine, C for cytosine, G for guanine, T for thymine, and U for uracil. Let's talk about nitrogenous bases. So with nitrogenous bases, they can be divided into 2 categories, purines and pyrimidines. Purines are nitrogenous bases with 2 rings, and they include adenine and guanine. These tend to be used as signaling molecules. For example, CAMP or cyclic AMP is a cyclic version of adenine. And the other type of nitrogenous bases are pyrimidines, and pyrimidines are nitrogenous bases with only 1 ring, and they include cytosine, thymine, and uracil. And so at the left here, you can see purines, adenine, and guanine, and on the right, you can see the pyrimidines, cytosine, thymine, and uracil. Importantly, you see thymine only in DNA, and you see uracil only in RNA. So you'll never see a uracil in a DNA molecule, and you'll never see a thymine in an RNA molecule. And that's an important distinguishing factor between DNA and RNA relative to nitrogenous bases. Importantly, you should know some nucleic acid definitions. A polynucleotide is generally 13 or more nucleotides long, and it's linked together by phosphodiester bonds. An oligonucleotide is a few nucleotides linked together by phosphodiester bonds. And here let's differentiate between oligomers and polymers. An oligomer stands for few similar repeating units. A polymer stands for many similar repeating units, and so they could be hundreds or millions of repeating units, as we could see in DNA. So when we say a polynucleotide, keep the definition of a polymer in mind. And when we say an oligonucleotide, keep the definition of an oligomer in mind. This in mind, a polynucleotide can be small. It could be simply 13 nucleotides long. However, oftentimes when we talk about polymers, we're talking about many, many more similar repeating units. The phosphodiester bonds found in DNA and RNA make up the backbone of DNA and RNA. And what phosphodiester bonds do is they link the 3-end carbon of one sugar molecule with the 5-end carbon of another sugar molecule. In other words, they link the nitrogenous bases together. So if it's DNA, it'll link the 3-end carbon of one sugar molecule, one deoxyribose, with the 5-end of another deoxyribose. For DNA to be packaged into the nucleus of a cell, it needs to be correctly and appropriately condensed. This is the function of chromatin. Chromatin is a complex of DNA and proteins, specifically histone proteins. It's found in eukaryotic cells and it's not found in prokaryotic cells. And the function of chromatin is to package DNA molecules into more condensed structures. The basic structural unit of chromatin is a nucleosome, and a nucleosome or chromatin is found only in eukaryotes. One nucleosome is basically a DNA helix wrapped twice around 8 histone proteins, or a histone octamer. Now these 8 histone proteins are really 4 different proteins duplicated. So a histone octamer is equal to 2 times 4 histone proteins. And these 4 histone proteins are H2A, H2B, histone 3, and histone 4. And you can take a look at the picture down to the right where it shows histone 3, 4, histone 2A, and 2B all binding together. The first step in the creation of the histone octamer is the binding of histone 3 and histone 4 together to form a dimer, and the binding of H2A and H2B to form another dimer. Then those dimers bind together to form a histone octamer. Importantly, you should know the difference between euchromatin and heterochromatin. Euchromatin is active, whereas heterochromatin is silent. What we mean by this is that euchromatin is accessible to transcription machinery. Heterochromatin, on the other hand, is inaccessible to transcription machinery. Both forms, euchromatin and heterochromatin, will be found in the nucleus. And you can distinguish between euchromatin and heterochromatin on electron microscopy. Euchromatin will be lightly packaged chromatin, whereas heterochromatin will be densely packaged chromatin. Let's talk about the Watson and Crick model of DNA. What Watson and Crick found out is that the DNA complex in our bodies is right-handed double helix. It's double-stranded DNA, and the strands are found anti-parallel to one another. That means that the 5' end of one DNA strand is parallel with the 3' end of the other. Watson and Crick also deduced that the strands are complementary to one another. That means that for a guanine nitrogenous base on one strand, you'll find a complementary cytosine base. And for a thymine on one strand, you will find an adenine complementary base. In other words, there is specific hydrogen bonding, so adenine and thymine bind together and guanine and cytosine bind together. There are 2 hydrogen bonds between adenine and thymine, and there are 3 hydrogen bonds found between guanine and cytosine, which makes guanine and cytosine stronger because they have 3 hydrogen bonds as opposed to 2. That means they more strongly bind to one another and less easily denatured. Another consequence of the complementarity of strands is that you will have the same number of purines and pyrimidines. This is because if adenine and guanine are the purines and thymine and cytosine are the pyrimidines, notice that for every purine you have a pyrimidine bound to it, and for every pyrimidine you have a purine bound to it. In other words, thymine is a pyrimidine and it binds to adenine, which is a purine. Similarly, cytosine is a pyrimidine and it binds to guanine, which is a purine. Because of this, because every purine binds to a pyrimidine and vice versa, you will have the same number of purines and pyrimidines. This is just a feature of the complementarity of the DNA strands. Also, Watson and Crick deduced that the bases or the nitrogenous bases are located interiorly within the helix, and the phosphate ribose backbone are located exteriorly to the helix. Let's talk about Rosalind Franklin. She made a crucial contribution to the discovery of the double helix structure. And what she deduced that the phosphate units are in the external part of the double helix. And so when we talk about the Watson and Crick model, we have to remember the contributions of Rosalind Franklin, who deduced this important fact.